Today we are blessed with a hardcore researcher and academician, Professor Dr. Satish Polshettiwar sir. Presently, he is working as Professor and Head Career Services and Placement at Dr. Vishwanath Karad, MIT World Peace University School of Pharmacy, Pune. Sir qualified GATE with 99 percentile and completed his PhD in Pharmaceutical Sciences. Sir is having more than 16 years of teaching and research experience, and he has in his name 3 patents, 4 authored textbook, 1 international award, 11 national awards, and he has published over 49 research papers in peer-reviewed national and international journals and more than 40 presentations at national and international conferences. Sir has been invited as a resource person for more than 40 workshops, conferences and online FTP. Sir is acting as a reviewer for more than 10 UGC approved journals, and Sir having YouTube channel with more than 10,000 followers. Sir has received more than 20 lakhs research grant and works as a pharma consultant for more than 14 industries. Not the least, recently he has given ICT tools to more than 2500 D-Pharmacy teachers. With this brief introduction, I request Dr. Satish Polshettiwar sir to start with the session.
Awesome morning to all my pharma educators and my pharma colleagues and my fellow participants. Indeed, it's a great pleasure. Before going to start this session, let me first, thanks my gratitudes towards the principal of this Sinhagad Institute of Pharmacy, Nare, KG Botra sir, and respected my guru, Dr. Kokre sir, my fellow colleagues, Dr. Amol sir, and other Vandana ma'am, Trupti ma'am, all Amruta ma'am for their untired efforts for making this event a more and more grand success. And I'm also thankful to them for inviting me as a resource person for very, it's a kind of need of our session that is advances in pharmaceutical research. It's the right time to understood all these challenges in front of all pharma educators and pharma researchers. So my topic of today's discussion, nothing but the very important, that is need of our repurposing of antibiotics and biosimilar by using pharmacophore approach and bioinformatics approach. It's a kind of revolution in research. This is a very need of a fascinating kind of research journey one has to start if you want to cope up with this challenge, because we are not thought about today's day that we are supposed to be remain at our home such a long duration, maybe from 1.5 months we are there at your homes, okay, completely locked down. So now it's a time to we cannot go outside, but time has come to go inside to see ourselves, to explore our knowledge, to explore to understand and enrich this kind of knowledge to make up ourselves for future ready. That is the very important kind of approach, a kind of called as need of hour at today's era. So that's why I've taken this topic to make you understood about this kind of tools, what are different fascinating tools, how we can integrate this IT, that is information technology, with your pharmaceutical research, and one can make our future bright. This is the kind of things that I'm going to cover today. Okay. Let us begin with this topic. First, disclaimer. This content of these slides are intended for educational purpose only, purely for the personal use. This presentation is based on publicly available information, that is OER, Open Educational Resources material. The view presented are the views of presenter, not necessarily to author, employer, organization. This is very important before going to start my this session. I am audible to all of you and I'm visible to all of you. Yeah, thank you very much for your comments. Let us begin with this topic. What are things I'm going to cover in this entire 1 hour session? We will make this session more and more interactive rather than monotonous way. So what are the things that I'm going to cover? We must know. So we all know it's a growing interest driven from shift from the classical to modern virtual method. Now we have to, all of you know, the one words in our late former president, Dr. Abdul Kalam, has already written in his book that in order to make our Indian empowerment, Indian population empowered, one has to one has to integrate this industry 4.0, or we call it as IT, that is information technology, with Indian talents. We have to integrate this, and one can make a better tomorrow, India's tomorrow. That is very, very important. Now we are witness with his vision of 2020, what he has dreamed and what he had written in his book. So IT plus IT is equal to IT square. That is the need of hour. So we must understood this concept ABCD. That is, we already yesterday we had a lecture of very, very wonderful lectures, a brainstorming session of Dr. Petesser, what he taught about ABCD. That is, A is augmented reality, artificial intelligence. B is blockchain. That is, we are using in supply chain management. Then C is cloud-based system. We are now rely on cloud-based system, virtual background, a virtual storage space. Then D is data science. We have big database like bioinformatics database. Today I'm going to explore you. So we have to only utilize and we have to retrieve this data for making more and more quality well-being. We can use it for your research purpose. Then I'll take you on a virtual tour on in silico pharmacophore approach. This is the kind of really fascinating tools to make you understood all how it is being done properly. Then a lab to clinic, translate to clinic to lab. Now this approach have been changed. Okay, so now we have to take the research from the clinic to lab. Okay, then it's a need of ours. One has to understood that. Then accelerate the drug development through repurposing and repositioning of existing molecules. This is my today's more focusing topic. How one can repurpose nowadays has come to utilize all the data and utilize all the information which is available on the web base. We have to use this data and we have to make these existing antibiotics. If you see the scenario of last 10 years, very few antibiotics have been appear in the markets. Why? Because industry are afraid. This R&D have approached to bring the new molecule into markets because of this drug resistance, a superbug. We are witness about that. Okay, this is the kind of third kind of threats to human being, public threats. A bioinformatics database are computational modelings through high throughput screening and high content screening. That is, I am going to highlight today how one can utilize these all bioinformatics tools. The next topic of interest that is biosimilar and genomic medicines. That is the future of pharmaceutical industry. Now everybody is looking after the biosimilar product. Okay, after the generic markets, we all of you know India is third position in the generic markets. We are the largest producer of generic medicine in the form of volume. Okay, and quantity also if you see. Then next area would be biosimilar. We have to make ourselves for this such area. Then recently PCI introduced this subject in our MPharm, that is called as CADD, computer-aided drug delivery system, and computer-aided drug design system that facilitate the rational drug design and development. Really, it is I really appreciate PCI for this introducing this such a nice and very a need of our subjects in our MPharm curriculum. Then very, very important that many of the MPharm students listening my lecture, this today's webinar, they are worried about how we can engage, how we can continue our research project. Those are in first year, and PhD student also during this pandemic situation. That is, I'm also going to highlight you how we can start your work, how we can make your proof of concept by using this virtual platform. Because now all of you know it is not possible to go into a lab, that is wet lab study. You would always believe on now the dry lab study. So there are a number of tools are there. Only thing is that we have to make aware about this kind of tools to your students. Okay, students must be aware. We have to explore this knowledge. All the big data. We have the huge data. Only thing is that you have to just synchronize it for your research. How we can use it, how we can retrieve it, and how we can analyze, and we can how we can interpret for your making the biological active components. So we are moving towards painful to painless. You all of you understood. This you all of you know for drug discovery and development process. Now the system has been changed. Everything have been changed. Now we move. We all of you understood the drug discovery process, drug design process started from either we use one approach that is receptor based, we call it as ligand based system. So ligand based is mostly nowadays targeting, receptor based mostly we are targeting. Okay, that is called as structure based drug design process. First you have to identify disease, any disease it could be, you can take, and then you can isolate the proteins. So it take normally if you go with this, it take 2 to 5 years. If you just identify the new proteins which is present in any organism, it could be bacteria, it could be viral, it could be fungi, it could be actinomycete, it could be protozoa, it could be other species also. So by using this in silico approach, by using pharmacophore approach, one can integrate, one can see the impact of your biological active receptors with your ligands, and one can screen it virtually without doing any kind of wet lab study. That is the beauty of this we call it in silico approach. Normally it take 2 to 5 years, but we can do it. We could make it within 2 or 3 months. One can get that proper ligands. One can screen. You all of you know drug discovery process. Then we move to the find drug effective against disease proteins. It required 2 to 5 years. Then preclinical testing, it takes about 1 to 3 years. Then we go for scale up and pilot plant study. Okay. Then we make a formulation. Then we apply for IND. Then we go to the human clinical trials. It took near about 2 to 10 years. We understood phase 1, phase 2, phase 3, phase 4 trials. It took a lot of years. Maximum drug get fail in phase 2 trials. Why? Because there are so many hurdles are there. Because the success rate of this drug discovery process, you all of you understood in your curriculum at very very less, 10% probability rate. Okay, that is we need to understood. Then we file NDA through our DCGI under CDSO. Okay, then we get a final approval for FDA and we can launch product into markets. That is the kind of discovery process. It takes near about 7 to 20 years to make to bring a molecule into markets. But if you use the silico approach, pharmacophore approach, one can reduce this timing. Till this preclinical testing, one can make till it this possible only 12 months only. Recently, I had just got one news in newspaper that one company, a Japanese-based company, have launched one fully artificial intelligence-based drug. Okay, within a 12 months. Normally, it took 5-6 years to brought the drug into clinical trials. But this is made possible because of these bioinformatics tools. Now people are trying to make this kind of process, this kind of fascinating tools to make our drug discovery process more and more effective and more. Normally if you go, it will very tedious process. It is time consuming. But if you go with this in silico approach, pharmaco approach, one can reduce all this drug discovery timing process. I hope you are getting all. This is very very important. One has to understood about antibiotic resistance, that is superbug, where the mortality and mortality of the particular drug against any particular test microorganism is very, very high, that we call as superbug, that could kill 10 million people in a year. This is we call as MDR is among the top 3 global threats. This is we call as a global threat to mankind. MDR, that is called as, you all of you know, microbial drug resistance, is among the top 3 global threats. India has one of the world's highest level of antibiotic resistance due to the overprescribing by medical professionals. This is the one reason. That's why we have to use this antibiotic very very rationally and very very judiciously in order to control, in order to avoid this drug resistance. Why all of you understood? Well, last 10 years, very few antibiotic appear in markets. Why? Because of drug resistance, superbug. That drug will not longer effective against that species. We understood escape molecule, a 6 very important molecule that we found a drug resistance. Excessive consumption of over-the-counter medicines. You all of you know 80% of antibiotics used in RTI, respiratory tract infections, are unnecessarily used. This has been observed. This has been proved, we call it as published in some literatures. 80% of the drug antibiotic which are used in respiratory tract infection are unnecessarily used. Means unnecessarily you are giving the more burden to your body, your immune system, because we all of our system have some bad bacteria and good bacteria. If you take antibiotic unnecessarily, it will kill a good bacteria also. It will it will just reduce our microbial flora, so our immunity will goes down. This one has to understood, and it will transfer the genes from one bacteria to another bacteria, and because of that the bacteria become resistant, and the resistant bacteria present in your system for 1 year. Entire 1 year, this bacteria will remain in your system. So one has to understood this. So because of over-the-counter medicine, over-prescription and all, we could get this, it induced the drug resistance. Abundant drug use in chicken and other animal food. That is also one of the major reason for this superbug. Why people are getting killed, and because of this. This has been published in Review of Antibacterial Resistance. History, if we just talk about the antibiotic history, the first antibiotic was discovered by Alexander Fleming in 1928, and later on he got a Nobel Prize in 1945. He himself, written in his book when he was discovered, he had discovered this antibiotic. It is not difficult to make microbes resistant to penicillin in the laboratory by exposing them to concentration not sufficient to kill them, and the same thing has occasionally happened in the body, and exposing his microbes to non-lethal quantities of the drug to make them resistant. Throughout the history, there has been continued battle between human being and this multitude of microorganisms that causes infection and disease. We all of you know now recently we have the COVID-19 outbreak. We are continuously battle with this microorganism. Now we have to cope up with this kind of challenge. We have to now rethink, redefine our education system, redefine our research system, research era, because post-COVID era would be totally different. the post-COVID era, what is going to be happen? Really, we are very fortunate that is we are part and parcel of this healthcare system. We are very fortunate because you people have a lot of opportunities in this field. We are witness about that. Okay, now the healthcare system is only working in the entire world. So AMR, that is antimicrobial resistance, continues to be public threat on global scale. If you just the global scale, as per WHO record and all, if you see, this AMR continue to be public threat. It's a kind of, one of the public threats on global scales. One has to understand, our body is, our environment is made up of ubiquitous with these microorganisms. Environment is ubiquitous, everywhere micro present. We have to always be tackled. Your immune system should be good for that. What is antibody resistance? All of you know antibody resistance. Okay, we are just reading and we are just knowing about all these things. Normally, what happens if in normal case, this bacteria, if antibiotic applied, it must kill or destroy the microorganism. That is the activity of antibody. Antibiotic are the substance derived from microorganism or chemically obtained that destroy and inhibit or we call as kill other microorganism at very low concentration because it is having MIC, minimum inhibitory concentration. We always believe on that. So normally it has to act like this. But what happens because of overprescription, because of over the counter, and because of this chicken and these people are using antibiotics for that, the normal species become resistance because of mutation. The genetic variability, genetic makeup is going to be changed. We know AGCT sequence, a genomic sequence. Now this genomic sequence is going to be changed. Because of this sequence, because of insertion, deletion, and this mutation, your bacteria will become resistant. It could be because of transduction, conjugation, translation. There are so many mechanisms out there. So we will discuss in detail of all these things. Then bacteria still survive because of this mutant species. And this continue to reproduce an offspring that resistance antibiotic used to treat it. So MDR is the top 3 global threat. That's why it is top 3 global threats. So we must understood this escape molecule, the 6 molecules, 6 microorganisms that is promising for making the antibiotic resistance. 80% of antibiotic used in RTR unnecessarily. That is the reason, cause we call as irrational use of antibiotics. Because of that, we could get this superbug on is. So what is antibiotic resistance? This resistance occur in bacteria. That is called as antibiotic resistance. It could be occur in endoparasites like flea and ticks. It could be in virus in case of antiviral agents. All of you know many antiviral agents produce resistance. Then it in fungi. All of you know fungi. Okay, many antifungal drugs produce resistance against this fungi. Then cancer cells. Many drugs like bleomycin and doxorubicin, which is used as an antibiotic in cancer cell, that also produce resistance. If we talk about journey from first antibiotic discovery to fully synthetic drugs to computer-aided drug developments, if you see, this is the golden era. In case of till 1960 was the golden era. Okay, till 1960. Now, after 1960, the first case was observed in tetracycline because of that efflux proteins which was found in the certain bacteria. Because of that, tetracycline will not, produce the resistance against the species S. aureus. What is called as a notorious species, one of the notorious species for making the drug resistance. Then biochemical process was started in 1970. Then increasing resistance. This is a linear, we could say. Then genomic high throughput screening was started in 1990. Then if you see, the drug discovery were completely, stopped in 2020. Very, very few molecules, very few companies have been brought the molecule into markets because of this superbug, because of this public threats. That is called a discovery voids. Look at this image, you will understood, okay, how many antibiotics start from your penicillin, then streptomycin, then first fully synthetic drug, that is called as sulfonamides, salvarsan, okay, that is all of you know, which is called as fully synthetic first molecule, and first antibiotic is penicillin. We all witness about all. There are so many reported cases of bacterial resistance according to this CDTPA, okay, reports. The first tetracycline I already saw, okay, in 1959, the first case observed against Shigella virus, in Staphylococcus, Streptococcus, Enterococcus. These are some escape molecules. They found the superbug. We all understood about the first generation, second generation, fourth generation, so third generation, fourth generation antibiotic, fifth generation still now we are using. After that, 2030 discovery voids because of these public threats. Now we have to redefine ourselves, redefine the research system, research, all the process and tools. Let us understood about mechanism of antibiotic resistance. It could be intrinsic, that is natural process. It could be acquired naturally because of lack of targets, innate influx proteins. You all of you know, or microorganisms because of efflux pumps, we have the free GP proteins. Because of that, drug is not taking out. Okay, they are coming out from the cells because of this innate process, efflux mechanism. This was observed in first case, that is tetracycline, against the one species, that is E. coli and P. aeruginosa. Then drug resistance could be the because of drug inactivations. The cephalosporin, in cephalosporin is get inactivated because of the cephalosporinase enzymes, which is present in the Klebsiella species. That is because of this drug inactivations naturally happen. Then biofilm formation, one of the called as very important mechanism in certain bacteria like E. coli and other species and S. aureus. Another way of antibiotic resistance is acquired. That is the 90% cases. This is because of genetic method, a genetic variability. That's why new era is coming. That is called as genomic medicines. Now the next era will be called as genomic medicine. I will be talking on that also. Then the genetic methods could be the chromosomal methods. It could be because of mutation. It is mainly occur in your TB species, that is Mycobacterium tuberculosis species, that is called as MSRA, methicillin resistance species. It could be because of extra chromosomal methods, plasmid. All of you understood the bacteria have the plasmids, that is called as extra chromosomal materials, which is present in the bacteria. Because of this plasmid, plasmid which carry genes resistance, are genes which is called as plasmid. Because of this gene carrying property, it could be because of a transduction, conjugation and transformation that we understood in microbiology. There are different way how the resistance is developed. One has to understood these fundamental aspects. Then and only we can work on docking study. Then and only we can utilize these bioinformatics tools for getting answer of all this solution. How we can repurpose the drugs. This is antibiotic target sites. Various targets we understood. Cell wall is one of the important target for these beta-lactamase enzymes and beta-lactam antibiotics like penicillin, ampicillin, and all. Another target area is DNA RNA. These fluoroquinolones mostly act on that. Then folate synthesis is also one of the way where drug is acting on that. Mostly clotrimazole. All of you know clotrimazole also nowadays produce a lot of superbug resistance against certain species. Then cell membrane, daptomycins, protein synthesis. Some antibiotics stop the protein synthesis like tetracycline, macrolide antibiotic, that is we called as your azithromycin's most popular drug. Then a resistance develop because of efflux. This is the major mechanism. If you see 30-40% bacteria getting produce the antibiotic resistance, induce resistance because of efflux mechanism. So one has to stop this efflux mechanism by using certain efflux inhibitors. We can use the phytoconstituents like curcumin, piperine. We could stop this efflux mechanism. Because we have to think about that. How one can control this antibiotic resistance. Then one can stop, or one can, antibiotic resistance is produced because of immunity and bypass. So one can use, immunomodulators. All of you know in viral, we have the witness about hydroxychloroquine. This has now been used for the prophylaxis of COVID-19 because of its immunomodulatory properties. This is the approach, how one can go with that, how one can repurpose the existing drug. So by studying, by getting this fundamental knowledge, one can start, your knowledge, your mind get triggered, and you can become critical thinker for getting a solution, for finding the solution, a prospective solution for this superbug. Now we know immunity bypass, because of that antibiotic resistance develop. We can work on that. We can give some immunity modulators. Along with the tetracycline. Maybe tetracycline may be effective against that particular species against later on. That one can find out, that one can take as a project, and one can give to project to your students. Then one can go with target modification. Mostly I am working on this. Today's topic of interest would be target modification. How we can modify targets, how we can prepare ligands, how we can use the target proteins. Because mostly all of you know, we deliver, proteins is very, very important. All of you know proteins and your ligands. One has to see the interaction between the protein and your ligands. One has to see the impact of this protein against this ligand, whether the ligand is acting on, you have 10 ligands, you have 1 proteins, 100 proteins. Just you have to screen virtually which proteins and which particular ligands acting on that particular existing target. Yeah, maybe new targets. Got it? There are some target sites for various resistant antibiotic, like in case of tetracycline, mostly target on this ribosomal point mutation. It could be, we all understood about this proteins, 50S and 30S. Mostly ribosomes have these 2 important proteins. In case of penicillin, mostly acting on modified penicillin binding proteins. Because of that, it produces resistance. Rifampicin, that is called as antitubercular drugs. All of you understood. Anti-TB mostly used in combination. A combination therapy is one of the way to tackle with this resistance. There are a number of ways are there. No, only thing is that you have to think about that in this way. How the antibody resistance is getting, because of target sites, structurally modified antibody target sites. So antibiotics normally bind to specific binding proteins on the bacterial cell. Now just look at this image, you'll understood. Now we have the bacterial cell. This is the bacterial cell. Now these are target sites. We have the 4 target sites. Now these are drug like antibiotics. The antibiotic normally acting on these target sites. That is we call as receptor site, a protein site we say. This is red color proteins. They are ligand, antibiotic called as ligands. Through this, it will acting on that, maybe through receptor sites, some attachment proteins and all. It will act on that, and it will show the the therapeutic activity. That is we expect from this. Okay, but what happens now is second image you could see over here. The antibiotics are no longer able to bind to modified binding proteins on the bacterial cell surface because now see the structure has been changed. The cell wall, because of maybe influx proteins, because of certain modification of structures, this is a target site actually, but now it has been changed. It is modified because of resistance, because of genetic transfer, because of a number of reasons are there. But 90% cases mostly happen in gene transfer. It could be transduction process, conjugation process, yeah, transformation process. In that, conjugation is most widely, prospective methods are we call as promising techniques to transfer the genes, resistant genes, to one bacteria to another bacteria, and that bacteria remain in our body for 1 year. Means that drug will not longer affect you. It will elicit that response, pharmacological response. Because of this change in site, it will block the binding. Now, see, it will not bind, it will remain because the drug will not enter, permeate in your cell so as to produce the effect. That is happening in, drug resistance. There's a need of ours. One has to understood investigating new uses of existing, because see, there are 2 ways. One, either you can go with the drug discovery process, new drug, by using de novo methods. Yeah, you can go with the existing molecules. But for drug discovery process, if you just think about drug discovery process, it's very, very costly, time consuming, and the time consuming and the pace required for timing is very, very high. So industry will not afford this kind of all kinds of for one drug you have to invest near about $500 million. So it's really difficult, and really all industry are not to afford all these expenditures, all these expenses. So that's why they have to rethink about your research also redefine ourselves, how one can repositioning this drug, how one can find out the new indication of existing drug, how one can indicate, how one can reposition, change the disease condition. Maybe one drug utilized for malaria, same drug we can use it for, you all witness about aspirin. So aspirin was introduced for NSAID, but now it is used for antiplatelet agents. What is that? That is nothing but a repurposing. We are repurposing the existing drug for new indication, new clinical indication. So many examples are there. We have recent example of hydroxychloroquine for COVID-19 prophylaxis because of its immunomodulated properties. So a number of we call examples are there. So discovering, developing new drugs need high cost, prolonged timeline, major roadblock for creating therapies. Drug repurposing is a safe, a successful pathway to speed up the novel drug discovery process and development process. You have to recognize the global health. So we have to take the help of this artificial intelligence. Yesterday we had a session of Dr. Petty sir, what he has introduced, artificial intelligence. So we have to use this artificial intelligence for making artificial wisdom to quality of well-being. That is very important. So artificial intelligence are there, but you have to use it wisely. You have to use very judiciously for making our health system more and more quality well-being. We are expecting quality in well-being. That is we are expecting. This, okay. So that you are supposed to use. Artificial intelligence are there. You have to just align with the software. What is AI? It's nothing but alignment of your brain with this software. Algorithm. It's a kind of algorithm. You have to integrate with this kind of technology, and we can bring a good solution for healthcare system. That is called artificial wisdom. Now it is converted to AI to AW. That is artificial wisdom. The need of our, so which particular drug we can take it for repositioning and repurposing? Mostly FDA approved drugs, and drugs that fail in clinical trials, which have detailed information on potential toxicity and formulation pharmacology. Because if you go with the new drug, then it's very difficult, because again you have to prove efficacy, safety, potency. But already it has been done by that innovator. Only thing is that you have to just repurposing existing drug. After patent expiry, all of you know patent is mostly granted for 20 years. After granting, it become generic molecules. So that drug we can use it for repurposing. Technical advancement in the informatics. We are really fortunate because of this technical advancement, AI technology, informatics, genomics, biological science account for the major success in drug repurposing, identifying secondary indication for existing drug. Drug repurposing is playing vital role in filling the gap in the discovery potential of antibiotics. Bacterial infection emerge as a ever increasing global public health threats by dint of multi-drug resistance of existing drugs. This raises the urgent need of development of new repurposing the existing antibiotic that can effectively fight against this MDRBI, that is called as multi-drug resistance bacterial infection. The present session describes the key role of drug repurposing in the development of antibiotics in combination therapy with the existing antibiotic. This is really, the need of our to rethink, redefine, rebuild up ourselves to in order to make ourselves for future ready to cope up with these challenges, because we don't know what is going to be happening in future. So there are some fascinating areas of research for all MPharm students, all PhD students, how we can go with these studies. Already we yesterday we had a discussion in detail about this kind of technology. So I'm not taking in touch detail about all this in depth. Okay, so I'm talking on only a repurposing drug, how we can repurpose it. Okay, because we have to make technology for better health. Now we are talking about cloud-based virtual screening. How one can use this IoT tool, that is Internet of Things, for exploration, for making our health better health. Pharmacogenomics is the another area of future medicines. Machine learning, deep learning, you have to make. Now we are talking about Industry 4.0. That is nothing but the digital revolution. We are witness about 1.0, 1.2, 3.0. Now we have 4.0. The new concept coming, iDoctor, iPharmacy. That is okay. Already we discussed about blockchain, cloud-based, and all. This is the from this image we understood what is the idea of repurposing. Now see, unutilized things we are using. You are giving some new kind of indication, new kind of use. That is we call it as repurposing. Repurpose. Okay, we have to use for repurpose purpose. That is we understood from this image. What are different strategy for repurposing? One has to understood. Okay, a repurposing drug could to key to fighting antibiotic resistance by using virtual discovery, using analysis tools. And I will take you on this virtual tool also, maybe after 5 minutes. I hope up till now we have been, you must have understood how this concept, drug discovery, and all this very important. What is in silico drug repositioning platform? That is nothing but the silico library. Virtual libraries are there. We have near about 3,000 database. If we just go with all these web-based tools, we'll find near about 3,000 database. We have PDB, PubMed, so many, n number of tools are there. So many softwares are there. One can utilize it. Only thing is that you have to just start practicing these tools. Know it, okay, understand it, then practice it and explore knowledge to others. That is the key for any learner, any pharma professions. Then it favor the drug screening, pharmacopoeia against new targets. We can use combination therapies. All of you know historically the many drugs we are getting through serendipitous discoveries, retrospective analysis of clinical trials. We all know the hydroxychloroquines, aspirins, minoxidil, metformin. We are now using for the breast cancer. Many drugs are there. The first drug which was introduced for cancer, girovidin, all of you must be aware, which was introduced in 64 for the cancer, approved by FDA for cancer treatment. But later on, after 10 decades, in 1985, the same drug was utilized for anti-HIV. Because of these bioinformatics tools, people got to know, because of this clinic to lab. Now we have to take the discovery from clinic to lab. One can understood. Now there's a need to develop this cloud-based system, a real-time monitoring system. How we can monitor, how we can get all this sensitivity of the antibiotics from different regions. Because our genes are 99.98% same, only 0.01% differ. That difference does matter. Okay, one has to understood this genetic variability. Our life start from DNA, RNA. We all of you understood dogma of life. DNA, RNA, then proteins, then we understood the cell, then tissue, then organ, then whole organs will develop, a whole system will develop. One has to understood the starting points. That is RNA and DNA. DNA is the starting points. Okay. There are few examples of repurposed drug. Like the first drug all of you know, which was repurposed, that is sildenafil. This question mostly all of you know. A sildenafil drug originally intended to treat hypertension, but repurposed for treatment of erectile dysfunction. Then another thalidomide, all of you know, thalidomide tragedy and all. Because of that, we could understood this PV, pharmacovigilance. Thalidomide originally intended for morning sickness, but repurposed for treatment of leprosy. That is another indication for this thalidomide drug, which was used for morning sickness. Then another buprenorphine, which was originally developed for depression, but now repurposed for smoking cessation. This is the different, chloroquine. Now, just now we have the witness with this very latest example, hydroxychloroquines. Okay. There are a number of drugs are there. Okay, I'll just not taking this. The only way to discover the limit of the possible is to go beyond them into the impossible, because impossible words say I'm possible. The thing is that we have to change our attitude, we have to adopt this new system, we have to change ourselves. That is very, very important. Okay, we have to think, your ideology should be changed now. We have to think, we have to redefine ourselves. Now, teacher role is going to be changed, act as a facilitator. We all of you know, not a content deliverer. Okay. Look at this, how we can use a pharmacophore approach for designing, and by using in silico approach. One example for that, one has to understood the fundamental aspects of mechanism. Now already I've told the in mostly 30-40% cases the drug resistance is developed because of efflux pump. What does it mean? Efflux means what happens? Taking out, drug is taking out. Now drug is coming inside. Now see, because of this proteins, drug is coming out. So this is because of this protein. This protein could be your PGP proteins. All of you understood in CADD. This PGP proteins. Now, if you want to avoid this phenomena, so what we can do? We can use some phytoconstituents. Okay, in your library, you have in silico library, we have database that I'm going to tell you, explore you. Now, you have to just think in this way. We can use this drug. You know, ideally, this drug is known for PGP inhibitors, well-known PGP inhibitors. If you add this drug along with your antibiotics, like curcumin, piperine, berberine, there are so many that you will be getting literature. On literature survey, you'll be finding, you'll be getting a number of these drugs. We can start screening of this drug against this drug, any antibody. You can choose ampicillin, those are outdated. You can take any kind of things, and we can just run your screening, virtual screening on this platform that is called as pharmacophore modeling by docking study, by using molecular docking process. And one can get a solution. Maybe this curcumin will give the solution for that ampicillin. Maybe ampicillin, we can use it again. This is the way how we can repurpose the existing molecule. What is the strategy for developing this? So various mechanisms of antibody resistance, including drug efflux, with the help of efflux pump, enzymatic modification, that is another way. Many antibiotics get degraded because of enzymes. So one can use some enzyme inhibitors over here. All of you know, mostly drug is acting as a inhibition mechanism or stimulation mechanism. So we can try this. We can, take the help of literature, take the help of database. We can go retrospective method. One can use clinic to lab, and one can get us, information from these doctors and all, and one can start working on that. And we can work on modification of target sites by using pharmacophore modeling. What is pharmacophore modeling? Nothing but the impact. We are going to check whether your proteins, whether your ligand is fixing or fitting on the proteins or not. Lock and key model like that. We have lock. You have to check the key whether your key is going to fit in the lock or not. That is called as pharmacophore modeling. So what is pharmacophore modeling? Lock and key concept, simple for your understanding. We have the lock. Now we have to choose the proper key to open that lock. That is called as nothing but a ligands. Ligand is your key, and you have the receptors. That is called as proteins, target proteins, or target proteins of interest. And we have ligand of interest. Just you try it. But it has to be done in a virtual lab. Now we cannot go to a wet lab because we don't know, maybe 6 months, 5 months, that we cannot predict. But we cannot start, we cannot wait and watch for that. We have to start our research. We have to start working on this kind of projects. We can give this project to your students. Student can work. Only thing is that you have to make them aware about this kind of tools. So you can learn from YouTube channels. I have created my own YouTube channel where we can learn all these things from first to last. Everything I have covered in that. Because in 1 hour, it is not possible to cover each and everything. This is things how we can, the representation of drug repurposing with the silent feature of both the process. Now you could see here how we can, if you normally it will take 10 to 17 years and 10% success rate over here. But here, if you go with this pharmacophore modality, okay, this process, one can make it 3 to 8 years. We can reduce the timing also. Only thing is that you have to validate the system. Very, very important. You have to work it very, very nicely. You have to validate the components. As you have to do it properly. You have to go with the clinical trial again properly. Okay, then one can get that product into markets. A classical to modern approach. What is this modern approach? We already discussed. Instead of going with the directly screening on this pharmacological approach by using forward pharmacology, we understood this phenotypic study and all drug discovery process. You have to go with the identification of proteins. You have to identify the proteins of interest by using a docking study, by using homology modeling, by using protein-protein interaction, machine learning, molecular dynamics. These tools are available. We have the tools. We understood 3D structure of these proteins, 3D structure of your ligands. Now, thing is that you have to utilize it, you explore it. Okay, by using these bioinformatics tools. It's nothing but the interdisciplinary field of computer science, mathematics, statistics, and biology, and one can make it into one single discipline, that is bioinformatics. These tools are fascinating tools nowadays to get involved in this kind of research. Then you can go with the functional activity in vivo after screening this. But this time, time required for this to this, it's near about 7 to 8 years. But we can make it to this maybe within 1 month, not 1 month, maybe 1 year. Okay, got it. This is a virtual screening, how we can do it by using this bioinformatics tool, immunoinformatics tools, pharmacogenomics, and pharmacoinformatics tools. So what is the drug repurposing workflow? How it has to be done? Okay, what is your strategy for working on this particular concept of repurposing and repositioning? So for that, first one has to get a list of approved drug which is approved by FDA and which is approved for clinical use. Then drug passed safety trials, that is phase 1 trial, but failing demonstrate efficacy in phase 2 for disease. Then drug replaced by better therapeutics, that is another approach. Then drug become generic due to patent expiry. These are the called as your interest things that are going to make it. That is called as drug. The first step for going forward with this drug repurposing. After that, then we can go with the serendipitous observation. Maybe you can get it from the clinicians and all. Data mining we can do. We can do virtual screening by using existing bioinformatics tools. We have n number of the data about proteins, ligands. We have the PDB websites where we can get all this information. On existing molecule we can work. It could be novel target you can find out. Okay, it is based on the observation that many drugs bind to multiple targets. This secondary target could be related to different disease or physiological condition. We can go with the known mechanism. If you have known the mechanism, new disease, it is based on the observation that several biological processes and signaling pathway are relevant for more than one disease. Hence, the same drug which inhibit biological process can exert effect on two different diseases. Then we can get that drug into that CKD. CKD is one example I have taken over here. Okay, for one particular molecules. Okay, then we call as preclinical, clinical studies. We can go and we can integrate into therapeutics. It reduces the risk, cost, time, and polypharmacy. Polypharmacy we all know, getting multiple things from multiple origins. This is a drug which is fully invented by artificial intelligence, that is AI technology. This is the first drug I could say, a DCP-118. Okay, the created medicines be used on women and first time on this OCD, observation compulsive disorder, by using the pharmacomodeling. The best example you could say. This is now normally drug development takes about 5 years to get into trials, but the AI drug took just 12 months. This is the beauty of breakthrough, innovation in the field of science, field of drug discovery process with the help of this IT tool, IoT tools, ICT tools. Now, you all of you know current medicine and future medicines. Yesterday we had a talk on that. Current medicine, if you just talk about that, one treatment fit all. But future medicine must be personalized medicines. Personalized medicine means now day will come, doctor will prescribe the medicine based on your genomic report. Maybe next year, maybe after 2 years, the doctor will ask you only your genomic reports. You have to just send the report by WhatsApp, by social media, and doctor will prescribe the medicines. That would be the era. Already even started in US, many areas. Okay, that is because of thanks to pharmacogenomics, pharmacoinformatics, and immunoinformatics and bioinformatics. That is called as personalized medicines. Because in personalized medicine there is no single side effect, adverse reaction. ADR is not coming into picture. We all know PV technology, that is pharmacovigilance, that is came into picture because of thalidomide tragedy. This is one of the best solution for that. Genomic medicines, that is the future of our pharmaceutical industries. One has to think about this line. That could be the era, maybe era will come. So we have to make our student on this particular area. That is called as biosimilar market, biosimilar products. A monoclonal antibody, we are looking after that. We all of you know many pharmaceutical industry working on this monoclonal antibody, which is more and more specific toward that antigens. Okay, specifically kill, specifically destroy that particular, disease conditions and all, target sites without producing any ADR effect, adverse drug reaction. There's no PV will come into pictures. So what is role of bioinformatics, which is coined by Pauline Hogg and Ben Hesper in 1970? It is nothing but the use of computational tools to discover new information in complex data set from one-dimensional information of DNA through two-dimensional information of RNA and three-dimensional information of protein to four-dimensional information of evolving listing system. with the help of bioinformatics, we could understood this COVID-19. You all of you know now COVID-19, COVID CoV2 is the higher version of advanced version of this COVID-1, that is SARS-1 and MERS. We all of you understood because of this bioinformatics tool only. One has to understood. You all of you know why it has made possible to understood all this is higher version because of this bioinformatics tool. Because already protein structure, everything we are known. Only thing is that you have to run this unknown proteins, we call as this COVID-19 proteins into your system, and you will get to know the evolution of this microorganism, how this microorganism have been evolved in the earth and this ecosystem. That is the beauty of this bioinformatics tool. This is the combinations we all know, integration of biology, computer science, IT to merge into single discipline, which involve biologists, pharmacists to collect molecular data, DNA, protein sequence, gene expression, and computer scientists because we have to analyze the data, we have to validate the data. So for that, we require mathematicians, statistics, develop the tools, software, algorithm. What is algorithm? Nothing but the integration of this, all these things. We require algorithm. We have to understand these algorithms to store and analyze the data because we cannot interpret all these things. We need to take the help of this computer scientist. Then that era, that single discipline come into picture, that is called as bioinformaticians. To study the biological questions by analyzing the molecular data. Now look at this one very important example I'm going to tell you. Then later on I will take you on a virtual tool. How we can modify, how we can do molecular docking and all. Now let us understood this is your bioinformatics database. This nothing but the virtual database. We have database like you all of computer database. Now your mobile phone also we have storage space, storage. Many things are there, many files are there, many images are there in your mobile phone. Similarly, in this database we have various nucleotides, DNA, RNA sequence, which is known one. Protein sequence also there. Many proteins are there. If you just know, it has been given in one literature, near about 15,000 drugs, 10 to about 20 molecules are there. We have near about 14,400 diseases are there. Among that, 5,000 treated disease and 7,000 are rare disease. And we have near about 19,000 genes which have been identified by various scientists. That is protein genes we call it. So this is a database already we have. Now thing is that you have to just take these known proteins like COVID-19, you all of new. We can take these proteins, we can take Ebola, we can take any kind of antibiotic protein also. We call as microbial proteins from S. aureus, E. coli. So that protein we can take and you can just add this protein into this database. That is called as virtual screening. Then what happens? This database will give the idea about the alignment and similarity in genomic sequence. Whether these known proteins or new proteins have similarity of genomic sequence with the existing data or not. Like we came to know about this COVID-19 too. It's a higher version of COVID-1 that is called as SARS and MERS. We understood. Okay. So that is we can get to know alignment and similarity in genomic sequence. We have the only thing is that we have to virtually screen it. Then this information is gathered from bioinformatics and we can get to know the evolution and history of organism. Any organism history evolution we get to know, we can understood with this kind of database. With the help of the genetic algorithm, with the help of this we call as help of this stochastic approach that we might have understood in your drug design process, structure-based drug design and ligand-based drug design. We must have understood about de novo theory, docking-based theory, QSR method, all these things. So these are target size, they are ligand size and molecular docking we can do. We can take a target protein, ligand we can take, known ligand, unknown ligand, okay, existing ligand, and we can do the molecular docking, whether we are this particular ligands going to fit with the target proteins or not. Lock and key simple. Again, we have to see the coordination, we have to see all descriptors, whether this particular ligands is going to fit, like key is going to fit it with the lock or not. We all of you lock. Then it's going to open the lock. Na? Your ultimate objective is to open the lock. So how to open the lock? For that, your lock should be fitted properly. It should be having that position, a confirmation that we understood. Confirmation, coordination, refinement of your structure, that is very, very needed, very, very important for drug design. We understood in QSR modeling, we understood in your pharmacophore modeling, prediction modeling, okay, that is drug design, ligand-based drug design. Computational powers, it is nothing but the computational powers. What is pharmacophore molecular modeling? Okay, then we will see this docking process, virtual library. I hope you all of you getting. Okay. What is pharmacophore modeling? Method. Use structure, that is SER, of ligands. Ligands nothing but the drugs molecule that is going to be target on your product of protein of interest. To propose the three-dimensional properties that must be present in molecules in order to for it possesses a biological activity of interest. It explains how structurally diverse ligands can bind to common receptor sites. That is the beauty of this. What is the outcome of this particular pharmacophore modeling? Structurally diverse ligand can bind to common receptor site. You lock and key again. We have 10 key and we have one lock. You have to match a particular key with that particular lock. So that is kind of the concept we should understood. The fundamental aspect is we must understood descriptors. Descriptors must be, you all of you know, as per Lipinski rule, whenever you are taking any drug, it has very, very important properties. That is first, the molecular weight. That is called a descriptors. The molecular weight should be less than 500. We understood in bioinformatics, being a formulator, being a drug design, being a pharmacist, we understood all. For making, for getting any drug inside the biological cell, okay, so it should follow the Lipinski rule. So what is Lipinski rule? It tells us about the drug permeation and drug absorption. Drug should be permeate and drug should absorb as per the BCS classification. We understood class 1, class 2, class 3, class 4. BCS class 1 is called as ideal class, having high permeability and high solubility. So it should be having, molecular weight less than 500. Next would be, it must be having a proper lipophilicity less than 5. That is log P value. That is partition coefficient must be less than 5. Then hydrogen bond acceptor should be not more than 10. Donor should be, that is hydrogen donor should be less than 5, not more than 5. Then it should be anionic, cationic, that is positive, negative charge must be there on that. It should be neutral, all of you know, which should be weak acid and weak base. You all of you know, drug should be always weak acid, weak base. It should be, ionized to get it solubilized and get it in the solution. But only unionized drug get absorbed in the system. Hydrophobic and hydrophobic property play vital role in that case. Aromatic rings, number of hydrogen atoms present. Okay, resolution also, your structure resolution. Then refining matter structures, conformation, coordination, all things we have to take into consideration while doing a pharmacophore modeling. These are fundamental aspects one has to understood. If you not understood this aspect, then it's very difficult to do docking process. For docking process, one has to understood all these, fundamental aspects. How one can make this particular, best fit model. That is the ultimate outcome. Lock and key again. Your key should be fitted on the lock properly. That is the ultimate objective of any kind of pharmacophore modeling. That is keep it in your minds. Okay. We use a zinc farmer. There are a number of softwares are there, but I'm going to tell you this very, very important and very, very, it's all free software. Another is SPDV, just to refine your drugs, your ligands, even proteins also. So zinc farmer is one molecular docking software. It's freely available. We can use it. It's a very, very good software and very user friendly. I'll click on that zinc farmer. Now see, once you click on that zinc farmer, just note down this website. Zinc farmer. Okay, sorry. Yeah, opening. I will open on other website. Okay, zinc farmer. Okay, just type on Google zinc farmer. I hope you can see over here. zincfarmer.csb.p.edu. This is a website freely available. There are a number of software. Gromax are there, Schrodinger are there, Atodoc are there, various software are there. But I found this software very eco-friendly and very, very user-friendly. Not eco, user-friendly. Eco also because all of you know it's economy. No need to pay anything. Yeah. Then once you open this, it will open this home window, that is home. Okay, it will appear. Then they will ask you search zinc. Search zinc means nothing but it's a zinc farmer is the software name. Okay, a kind of software tools which is designed by the scientist. Okay. Then you just click on zinc farmer. So for docking purpose, what you required, we should have a proper ligands. You should have the, because you have to repurpose the existing drug. No? So that you should have your ligand molecule. You should have your protein of interest. It could be 5 proteins, 10 proteins. What are the proteins we have identified? That proteins you have to take it into consideration, and you have to take different molecules. You have to make the structure of different drugs. Okay, then you can just run the, virtually we can see the impact. Like lock and key, we have the 10 lock, we have 10 keys, and we have to just find out which particular key is going to be fitted with my lock. That is kind of docking process. That is called as docking process. Okay, just click on search zinc. I think I'm running with time plus, but I will try to finish it. Don't worry. Okay, I'll not directly see. There are different way. The first thing is you have to load receptor over here. Then you have to load features. What features means what ligand you are going to add over here. Both options are there. Now I'm taking directly the existing molecule, okay, which is already made, okay, because it will take time to, docking process require 2 hours for one docking for one molecule. It's not possible to make it like I'm taking the existing molecule which is already made. Like I'm taking the kinase inhibitors, okay, for example, okay, which is using NSAID drugs. Okay, these are kinase inhibitors. Now see, you could see here structures over here. Now see, it will appear over here. Okay. Now see, this is the 3D structure. Now you can see the 3D structure of these proteins. You can see these are the protein structures, okay, in 3D form. The thing is that whenever you are making docky, always look at these descriptors. How many descriptors you have to add? You have to remove the unnecessarily hydrogen atoms. You have to refine the structures. You have to use. You have to remove the cofactors, which is used for recrystallization under XRD study. Okay, that is very very important. These are drug molecules. That is we call it ligands. It could be antibiotic. It could be biosimilar. Okay, this ligand molecule you have to see whether this molecule is going to fit it with your lock and key again. Again, whether you have to see the conformation, coordination, whether this drug molecule is going to fit it with this kind of protein structures. Then descriptors, we could find here different descriptors over here. Hydrogen donor, I already told you. Hydrogen acceptors, now see it is minus, this is minus 3.8. Hydrogen acceptor is less than 10. Hydrophobic properties, you can see all the properties over here. We can also manage this, okay, depending on your product of interest. Hydrophobicity, hydrophilicity, that's very very important, all the descriptors. Okay, we can add a feature also. Many feature we can add, okay, and we can run the query after that. By you have to see the, because there are n number of molecule present in this particular software. Only you have to just run the query. Okay, now see you'll be getting a number of hits. How many? Because you have, for example, 1 million proteins and 1 million your drugs. Yeah, maybe 5 drugs. You have to see these 5 drugs, 5 ligands is going to be hit with which particular molecules. You'll be getting that idea. Now see this uploading query. It will take some times. So it will appear over hits, number of hits. Then you can choose that hits. Now see number of molecules present over here. So likewise you can go it over here. Okay, got it? This is the way how we can do it this. Because of time constraint, I could not continue with this. Okay. Then we can get PDBs. This is one in order to make your proteins in PDB structures. We can visit this website, that is rccb.org, where you'll be getting the lattice proteins. Now see, you could see a lattice proteins. This is a month molecule May. You can upload if you have known proteins, known ligands. You can also deposit. The option is there. You can deposit. This is a database only. There are about 3,000 databases are there. We can deposit your database, your proteins, your all these things. Now see, this is a recent advance. This is the May molecule of month. This is the somebody has submitted this proteins. We can take these proteins. We can download this protein also. Options are there for downloading purpose. Okay, we can search over here. Like I'm searching on this IFAA. This is one Ebola proteins. Okay, which is act on Ebola. You'll be getting the proteins over here. Now see. Yeah, it will open. Yeah, now see. Okay, it's not appearing, but you can take any this VP proteins, VP30, also Ebola proteins. Now you can see a number of proteins which is act on Ebola virus, Ebola mosaic virus. Now you can see 1YCX. You can take this 3D structure, you can download it in this different format. Download option is there. You can download it, okay, in PDB format or Java format. And we can take it into your Zinc format later on. Now they're asking you to save in your library. You can save it later. You can save it in your file and you can use it for docking process. Then number of proteins are there. See, you can get the protein structure over here. I'll coming to this slides now. Okay. Now next would be protein ligand docking. We can go with the search algorithm. Yeah, scoring function. We have to score it. The affinity should be minus 8. If your binding energy is less, then it will have more impact on your drug molecule or your receptor molecules. We always expect a lowest energy, a GIF energy, that is delta G. All of you know. We mostly take it into consideration this intrinsic free energy, because whenever drug acting on your receptor site, it has a high energy, but whenever impact on your receptor, it will lower the energy. We measure the lowest energy level, a kinetic energy. That is the that is called as scoring function. We calculate the scores of binding affinity on particular poles. Different softwares are there for molecular docking. We can take the docs. Gold is commercial. Okay, we can Schrodinger, VLab Science also there. There are Gromax are there. N number of softwares are there. But these are the Zinc Farmer I found very very friendly and very useful for simple projects. Only thing is that docking calculation scoring is very very important. You have to always take into consideration, either going with one, you always try to 2 or 3 in order to validate your this particular docking process. I would suggest don't use only one. Always try with the gold and AutoDocs. If you try with the AutoDoc, if you're getting one molecule, always try to another software in order to validate this particular scoring process and reprocess this. There is a docking process, normal process and reverse docking. Sorry, What is normal docking? We take the protein of interest, we put it into your database, chemical database, and we get the possible binding ligands like lock and key, like that. Okay. Then reverse docking, what we do? We take active components of existing drug for repurposing your existing drug. We put this drug, incorporate into drug in this library that is called as the a protein target database. Then we'll find the possible binding proteins. This is the beauty how one can go with the docking process and reverse docking process. This is a molecular docking process. How one can go with the HIV proteins. We have the ligands, we have the HIV proteins, docking scoring, we can do it. Okay, we can see the pose of this all the molecules. Now see, this is having highest pose to 6.84. Then this is the ligand 2, which is promising because minus, more than -8 always better. It should have lowest energy always. Then and only we can say it's a promising molecule for further drug discovery, further clinical trials. We can go with the biological activity further. This molecule, we found a promising molecule in all this molecule. Okay, all this key and lock also, we have lock and we key. We have to find out this key we found very nice, very promising, very potential. What are biological biosimilar products? We are aware about biological. Now see, the era will come maybe next 2 months, near about 20 blockbuster biosimilar products, biological products are going to be patent expire. Now after generic market, the new market coming in the, in front of all pharmaceutical industry, that is biosimilar. You already many industries have started, Shanta Biotech, which is taken by Sanofi, you are Dr. Reddy's, so many companies, Pfizer, all companies are now moving, forward for making of these biosimilar markets. Not lot of opportunity for young farm students in this era. Now we have to always think about these biosimilar products. So what is biosimilar products? Biosimilar product means biologically we call it highly similar, but that is not identical. Like generic market, in generic product we making the similar copy, but here in biological it's not a similar copy. It's a highly similar but not identical. It's somewhat, chemically they are inactive components. They're highly similar with the reference product in terms of safety, purity, and potency, but may have a minor difference. Your monoclonal antibody, your vaccines are called as nothing but the biosimilar products. Biological similar, name indicate biological similar from small molecule to larger molecules. We are now moving toward the larger molecules in order to prevent, in order to make it available for common people in affordable rates. That is called as biosimilar. Because we all expect the affordable price and quality medicines. Like generic, we all know generics are having quality as well as affordable price. Anyone can afford that price for generic market, generic product. Like similar, biosimilar, very costly. If you go with the biosimilar product, very costly. Now we have to think about this biosimilar product. Biological are very costly. Vaccines, monoclonal antibody are very costly against this cancer therapy and all. One has to always think about this line. This is one example how one can get the bound X-ray structure and unbound X-ray structure, how it looks. Rigid docking and flexible dockings are there. Then one of the example of turmeric, it has been observed against the Ebola virus. Okay, because of the virtual screening, it has been made possible to explore the effect of turmeric. You all of curcumin is the very promising agents in the curcuma longa. You are 77% curcuminides and only 2 to 6% curcumin present. That curcumin have a promising properties against various viruses and various kind of antibacterial activities. Various software we can use. Already we told molecular docking can be done by docs, JDocs. Because of time constraint, I really sorry for that. I could not complete my entire session, but I will try my best level to finish within next 5 minutes. I think it is okay for coordinators, organizing team. Yeah, please. Shall we continue for next 5-10 minutes? Yeah, should we stop?
Yes, sir. Continue for
really interesting
5 minutes.
That so many things are there. Okay, molecular docking can be done by searchable function, a scoring function. Okay, so it could be target flexibility, ligand sampling. We'll take it in detail later on. Okay, so I can share my own videos where we could find all these things. Reverse vaccinology also another area of biosimilar product development. Instead of going with the whole organism, now scientists are trying on the specific epitopes. So what is epitopes? Nothing but again key, again your antigen. Instead of taking whole organism, now we are moving toward the painful to painless. So if you take this, it will painful because whole microorganism you're taking. Unwanted parts are there, because we want only this part which is having the immunogenic property. But if you take whole microorganism, then it produce the painful. It causes, you all of whenever take a vaccine, it causes the at some adverse reaction, some kind of response, immunological response. But if you take this kind of vaccines, this kind of protein only, now you all of surface proteins are being used. Now you all of rotavirus vaccines. Rotavirus vaccines which is used against diarrhea, which use this surface proteins only instead of taking all these things. Now, thing is that only you have to recognize these proteins on this particular antigen. Now, these antigens are there, which is having multiple epitopes. Like COVID-19, if you take example of COVID-19, it has a multiple epitopes, multiple antigens. That's why all scientific community are getting failed to develop the vaccines in very stipulated time because of multiple epitopes, multiple antigens. One has to identify, recognize these epitopic sites because antibody have paratope site. Paratope is your nothing but the your lock, and this antigen is nothing but the key. In order to open this lock, you need to have a proper key. No? If you have multiple key, then it's very difficult to open all this problem. That is the thing one has to understand. If you go with this reverse vaccinology by taking into these antigens, then one can develop this very, very promising, biosimilar products. Now, painful to painless vaccine. Now, all of you know why this rotavirus vaccines are painless, because of you are taking only this small antigens. We call it as very specific portion of these unwanted parts are there. We are removing the unwanted parts, so it becomes painless. That is the concept of painful to painless. Now you must understand. This is reverse vaccinology, again, same process. Okay, already I have explained that is the monoclonal antibody we can develop, vaccine we can develop through that. This is the one has to understood the mechanism of drug absorption. So we can understood what are different proteins, influx transporter will be affecting on your antibody resistance. We already understood about that PGP proteins, ABCT, OCT, ASBT, OCTN, organic, anisotropic. There are so many proteins are there, efflux and influx transporters. PGP is one of the influx transporter. BCRP is there, breast cancer, resistance proteins are there. So many proteins are there, efflux and influx. One has to understood that. We have some intentional metabolism enzymes also there that also could play a role in antibody resistance. So many things are there. So we have to understood all this bioinformatics, cheminformatics, neuroinformatics, genoinformatics, metaboloinformatics, then health informatics. Okay, this if you combine, integrate all these branches, then one can get a very good solution for this, kind of public threats that is we call as. There are different database. Okay, just to take a snapshot of that. If you want to know about protein sequence, you can go with NCBI. If you want to know about enzyme sequence, you can go with Chem. PDB is most widely used for this antibody resistance. There are a number of databases are there. Some are sequence, some are structural, some are functional. Okay, a structural database is most widely used, and functional also we use because we need to understand genomic sequence, RNA sequence, DNA sequence, protein expression, and all these things. There are three important biological databases: primary, secondary, specialized. Okay, secondary mostly used because you all of you know it is very highly curated, highly a pragmatic and practical solution that will provide. Yeah, there are n number of databases are there. It's vary in size, quality. It have to give the comprehensive solution for our researchers. It give accuracy, is up to date. Just now we saying COVID-19 already up to date in that PDB genes, PDB software. It has a good interface, batch search and download. You can many things we can do it. It's freely available. Okay, only thing you have to explore it. These are NCBI websites. These are the different websites: PubMed, DDBGI, PIR, Protein Information Resources. Then Ensemble genomic browsers also there to get this genomic project information. We have UCSC genomic browsers, okay, for downloadable and genomic browsers, okay. A National Center for Biological, called as information centers are there where we can get all the information of genetic informations. We can submit it, we can download it, we can learn it, we can analyze it, we can research it, we can develop. All these things we can do it with the NCBI. Multiple things are there. Okay, already we are running with the times, so I should, yeah. There are a number of research also done on this molecular docking and all these things. Okay, I also published 1 or 2 articles on that. Okay, gene banks also there. Okay, yeah, I'll just 2 minutes. We'll finish this session. Okay. I think I have done all the informative informations. Okay, in order to go with the docking process. My aim was to just to make you ignite your minds to move forward against this line. That was only my aim. Okay, because in 1 hour it is not possible to get in touch with, get in depth about all this entire docking process. For one molecule docking required 2-3 hours. So now I have covered each and everything. It's the kind of ignition, a kind of triggering process. Uniprotes also there we have. Okay, n number of websites are there. Okay. So only thing is you have to use it properly. Structural database also there we have. Okay. I'm not taking this because of time constraint. This is the structure you can get each and every information about that 3D proteins. We can download also. Okay. in the form of PDB format, that is called as standard database. You can get it into the Java format also. In order to combat drug resistance, no action today, no cure tomorrow. If you're not acting on that, acting on this level as per WHO, no cure tomorrow. The conclusion, finally, I concludes, the antimicrobial resistance threaten many aspects of human activity, including medicine, agriculture, that could lead to more death than cancer. Drug repurposing is very unnecessarily and pragmatic approach, very, very realistic and practical approach to increasingly identify the antimicrobial activities in several approved drugs. This required a cooperation and goodwill of big pharmaceutical companies for its successful transition to clinic, and can positively impact the identification of new antimicrobial targeting the drug-resistant pathogen, a highly unmet need of healthcare community globally. The drug repurposing strategy is widely used as an alternative approach to drug development, since it lowers the risk of safety, toxicity, and it's at the same time save the millions of dollars worth of pharmaceutical R&D. Over the decades, many drugs have been repositioned, licensed for use in alternate indication, and many more repurposed drugs are currently at the phase 2, phase 3 clinical trial. The drug repositioning approach is the simple yet powerful strategy to fuel pharmaceutical research, streamline the drug discovery process. A computational modeling would be one of the key indicators for repurposing of drug. A cloud-based service need to be developed for analysis and real-time monitoring of antibiotic sensitivity. One has to think about this line, how one can make a cloud-based sensitivity, cloud-based services to link all doctors, all the clinicians, okay, to collect all the data, because one drug is effective against particular person, may not be effective against another person. That information we don't have. So we need to act on this line. For that, these IoT tools, Internet of Tools, could be one of the the breakthrough innovation, breakthrough medicines, we can say, fascinating tools to make and cope up with this challenge. The virtual innovation lab to radically transform how future medicine health services are developed and delivered. This is very, very important. Finally, this is the conclusion from my sides. Post-COVID era would be totally changed. Healthcare industry is going to be benefited. We are very fortunate to part of this particular data science, herbal medicines, and all this is going to be benefited. There are references for all these my slides. This contents of these slides intended for educational purpose, purely for personal use. Thank you very much. In order to mold yourself, you have to be intellectually sharp, mentally alert, spiritually elevated, and physically fit. Final statement: use antibiotic rationally, judiciously. If you know action today, no cure tomorrow. Thank you once again to organizing team. Stay at home and take care. Yeah, I'll take this thing to on organizing team now. Thank you for listening me. I think I had made it very, very, lengthy somewhat. Okay, but okay. Thank you for bearing with me. Yeah.
Thank you very much, sir. It was very good presentation. I really liked the image of repurposing. It explained itself. So session was very excellent, informative, and the language for session was very simple.
Yeah. Thank you for your.
And thanks for the live demo of the zinc farmer. It was very nice.
I could not make it because of time. Okay, but I tried my.
We will take another session for that also.
Definitely, madam. Because see, 1 hour is not possible to cover each and everything.
Yes.
Okay? Yeah.
There are number of questions, but due to the time restriction.
No, you can take, madam. Sure.
Okay. Then we will
you can
discuss.
We will address all these things.
Okay. So first question from our student, that is Swanan Kulkarni. So Swanan, you ask questions, sir.
Yes, ma'am. Good morning, sir. Good morning.
Good morning, ma'am. Good morning, sir. Yeah, nice to talk.
Actually, I'm Swanand Kulkarni. I'm pursuing my master's in pharmacy in M.Pharm Medicinal Chemistry from Central University of Punjab. Sir, actually, I have a question related to the crisis that we have related to the irrational prescription of antibiotics.
Yeah.
I have seen so many doctors prescribing amoxicillin and other penicillin, cephalosporins for some small issues such as throat infection and common cold fever.
That I alluded to 80% of this unused. Yeah.
Yes. Being a healthcare professional, being a pharmacist, how can we contribute to this crisis, sir?
Yeah, very, very fascinating and very, very really, one of the need of our questions you have been asked to me. I really appreciate for that. See, being a pharmacist, now thing is that we have to integrate with the IT. Already I told IT plus IT, Indian talents. We have to take the IT integrated with your system. One can make a cloud-based system This cloud-based system will monitor real-time monitoring system. Maybe you can give some kind of devices to the patients, and one can give to the doctors also. Doctor will just putting that all information into that cloud-based system, so one can identify that person is sensitive to this antibiotic, some may be resistant to antibiotic. This real-time monitoring will be helpful to make our this kind of public threat problem, and one can give solution. Only thing you have to go in this line. It's a big project. I really am fortunate I'm a part of that project, because I have taken into, as MIT, we have taken this project at large. I am a part of that. Really, I'm fortunate for that. I hope you got my answer. Yeah.
Yes, sir. But on personal level, how can we combat with this situation?
First level, sir, see what happens. Only thing is you have to use it judiciously. You have to, rational use is very important. You have to always make aware about this thing to clinicians. Being pharmacist, you have to counsel. Counseling is very important. Patient counseling. You have to always ask the patient, always ask the public to take this medicine very rationally, because many people, if you're having throat infection, they directly take this drug unnecessarily, improving the burden, increasing your burden on your system. So the thing is that the pharmacist role is very important. Now you are the part of this healthcare system. Now it's our duty to make aware this campaign you can take, like the campaign you can take. You can go with this campaign maybe in the form of NGO and all, and you can work on that. This line definitely at start area. Definitely it will be the fascinating. It would be the revolutionary things. It will come.
Yes. Thank you,
Thank
sir.
you.
Thank you so much.
I hope
Okay,
you got satisfied.
Yes.
Yeah.
Yes, sir. Yes, sir.
Thank you,
Please.
Swanand. Thank you, sir. The
Yes,
second
thank you.
question from the Madhura Datar, that is, does the repurposing of medicine produce any adverse effect in body?
That need to be validated, ma'am. Yeah, because see, without adverse effect, all of you know, drug is called. All of you know, we understood bala phasia in pharmacy symbol. We pharmacists make the toxic drug, toxic molecule into active molecule. Every drug has side effects. Only thing is that we have to validate it. You have to just minimize the safety, efficacy, safety, toxicity level. We can bring down. It's possible to bring down zero. If zero is bringing, means the drug is not active. Okay, so always it should have some side effect. Only you have to think, you have to minimize, you have to mitigate all these things. You have to minimize the side effect by using some validation tools. Every drug has side effects, but only thing you have to work on that. You have to just you have to work on these toxicological parameters.
Okay.
You got
Thank you
my
very
this
much.
thing.
Yes, we got. Thank you very
Yeah,
much, sir, for answering the question.
yeah.
There are a number of questions, but due to time, we will stop the
Yeah,
question answer.
one or two questions. I'll be available on WhatsApp also. If anyone have any
Okay.
queries, you can just discuss on my WhatsApp. No problem. I can
WhatsApp
share.
or otherwise mail to me or sir directly.
Yeah. Coordinator also there, organizing team. You can contact through that.
Yes. Thank you very much, sir, for sparing your valuable time. Thank you very much from Sinhagad Technical Institute of Pharmacy, Narai. Sinhagad Institute of Pharmacy, Narai. Sir, should we conclude the session?
Yes, it's my side. Okay.
Okay. So I declare
Thanks
the
to all.
session is okay. I declare the session is over. Stay home, stay safe. We will meet tomorrow we will meet today evening at 4 o'clock. So thank you very much.
Thank you, madam. Thank you once again to all.
Thank you.